Everyone, myself Pallavi Sharma. We are going to study some important question of the chapter cell cycle and cell division. The first question is number of meiotic division required to produce 100 macrospore in a angiosperm. So macrospore refers to the ovum or the megaspore. For production of one ovum, we need one megaspore mother cell because each megaspore mother cell undergoes meiotic division and produce one functional ovum at one time. So for the production of 100 macrospore, we need 100 megaspore or macrospore mother cell is required. So for the production of 100 meiotic division is required. That is why B is the correct answer. Next question is for studying meiosis, most suitable material. So root tip, shoot tips, mature leaf and young floral buds, all of these are responsible or require mitosis to increase the number of their cell, to increase the cell length, as well as to increase the activity. While young anther cells and the testes of grasshopper, these contain the germinal epithelium or the germinal cell, which are responsible for the production of pollen grain or sperms in their body. That is why D is the correct answer. Next question is in which stage size of nucleus increases? As we know, the prophase, each stage of prophase representing cell division stage, which is M phase. I phase represent interphase of cycle, an interphase containing G1, G2 and S. During S phase, specifically the S phase is synthetic phase and it is responsible for increase in nuclear content. So the correct answer is A, because the size of nucleus will increase during the I phase, which is interphase. Next question is which is called direct cell division. So mitosis, meiosis, your odd cell divisions, this is not the correct answer. Correct answer is amitosis, because amitosis is also known as direct cell division. The reason behind is that it involves cell division without chromosome and spindle formation. That is why the correct answer is B, amitosis. Next question is if a wheat plant has a minimum number of meiotic division required to obtain 100 grain. So we have studied that megaspore mother cell produces one functional megaspore at or after one meiotic division. So for the production of 100 ovum, we need 100 meiotic division. But this is not in the case of pollens, because seeds is the final product of fertilization and this is formed by the fusion of ovum and pollen, so that will form later on seed. So pollen are produced in tetrad form. Tetrad means 4 cell and all of them are functional. So for the production of 100 pollens, we need like 25 pollen mother cell will divide 25 times to produce 100 pollens. So the answer will be 100 plus 25. So 125 meiotic divisions are required for the production of 100 grains. Next question is, in metaphase 1, each centromere of a chromosome, how many chromosomal fiber or the tractile fiber arise toward the pole? So if we study the structure of the chromosome, here centromere, two arms, two chromatids is attached to one centromere, which is covered by C-shaped or disc-shaped structure known as kinetochore, attached to spindle fiber. One side to this side is attached to one pole and this side is attached to the another pole. So if we are talking about the one pole, each pole contain one tractile fiber. So the correct answer for this is 1, because for each pole there is one tractile fiber. Next is disjunction, separation of chromatid at anaphase. So disjunction, if we are talking about disjunction of sister chromatid, so that means we are talking about anaphase 2 in meiosis 2. In this case, what will happen? The homologous chromosome, which we have studied that separated during anaphase 1, will be pulled in the opposite direction. Toward this direction they will be pulled. So what will happen? This results in something, this kind of structure, where this is the position of centromere. So centromere is pulled first, which is followed by chromatid. That means the centromere is moving earlier and the chromatids are trailing behind. So the correct answer is A, which is representing the centromere move first and arms trail behind. Okay. Next question is, chromosome at anaphase are of various shape depend upon the position of centromere. It is J-shaped when the centromere is. So we are studying everything. For the metacentric, the shape of chromosome is V shape. If we are talking about telocentric, it is I shape. If we are talking about acrocentric, this is J shape. And if we are talking about submetacentric, this is L shape. So the correct answer is C, because they are asking about J shape, and J shape chromosome are found in like acrocentric chromosome. Next is, in egg cell has 5 pg of DNA, and in the DNA in the nucleus, how much amount of DNA will be in this animal cell at the end of G2 phase? See, When a cell just came out of the mitotic phase or the division phase, it contained 5 picogram of DNA. Okay. Now this will enter into the G1 phase. It will still remain 5 picogram. But if we move another step, so there will be increase in DNA because this will enter into the S phase. So the DNA here it will increase because we have studied DNA changes from 2C to 4C. It is already 1C. So it will increases more time. That means it will increases about 20 picogram in S phase. So he asked about in G2 stage. So in G2 stage this will be 20 picogram. So the correct answer is D. Because in S stage synthesis of DNA takes place. Moving on to the next question. Next question is, if by successive division of a bacterium every minute a small test tube is filled up in 1 hour, the lower half of the test tube will be filled up in how much time? See, this is an experiment. We have given a test tube whose upper half, which is this, so it is filled in, the time is given, this is filled in 1 hour, that means 60 minutes. For filling, 60 minutes was required to fill this upper half. Then we have to find out the lower half. Okay, so what was the doubling time for that bacteria is 1 minute. That means generation time of that bacteria is 60 seconds. Okay, upper half is filled in 60 minutes. That means for the lower half they need 60 minutes minus 1. Why we have written 1? Because at the position of half is already filled. That is why answer is 59 minutes. Answer is C. Next, moving on to the next question. Colchicine, granosine and vinblastine inhibit cell division, bring polyploidy. First of all of these are cellular poisons. Second thing is polyploidy. So, beta polyploidy is having more than one set of or more than two set of DNA or chromosome. If a cell having more than 2 set of DNA, that is known as polyploid. For example, if we are talking about 3N, we are talking about 4N, we are talking about 5N, all of these are polyploid cell. So, what is the function of these cellular poisons? They inhibit spindle formation. Cellular poison inhibit, this is the sign of inhibition, they inhibit microtubule polymerization. And if microtubules are not polymerizing, that means they will not move to the next stage, they will stay at one stage only, and that is why A is the correct answer. They inhibit spindle fiber formation because spindle fiber are made up of microtubule. Moving on to the next question. An organism having 22 pairs of chromosomes celled at the end of meiosis 1 will have, if we are talking about 22 pairs, that means this organism having total 44 chromosomes. And we are talking about meiosis 1. So meiosis 1 is already a reductional division. And if an organism having 44 chromosomes, after meiosis 1, these get reduced or these get halved. That is why answer is 22 chromosome after meiosis 1, each of the cell having 22 chromosome. Answer is B. Next question is, how many times a cell has to divide mitotically to produce 128 cells? So for this we have to use a formula, that means that is 2 की power n, and n is number of mitotic division. If we put value like 2 की power 7, it will give us value of 128, or we can even calculate the n by putting simply into the formula. So if 2 की power 7 gives us 128, so answer will be 7. Why not 64? Why not 126? Like they have asked about a single cell, a cell, so that is why we are answering, or we are assuming that is a single cell is going to divide 7 times or 7 generation to produce total 128 cell. That is why answer is D is correct. Next is, at dikinesis, meiocytes have 10 pair of chromosome. The chromosome number expected in each nucleus is therefore, so the first option is 10 at the end of division 1 and 5 at the end of division 2. So see, they have given the keyword which is 10 pair. If they have mentioned 10 pair, that means they have total 20 chromosome in a cell. After meiosis 1, as we know that meiosis 1 is reductional division, after meiosis 1, each cell will be having 10 chromosome. And if we are talking about meiosis 2, which is equational division, then both of the cell having 10 chromatids or 10 chromosome. So B answer is correct, that 10 at the end of division and 10 at the end of second division. Next question is, chromosome appear, we did the same question earlier, that chromosome appear L shape. So this is the direct answer. Submetacentric chromosome appear L shape. So the answer is D. Next question is, starting with bacterium that divide once in a half hour, how many bacteria are formed after 24 hours of growth? So what we have given is doubling time, which is given 30 minutes. And they have also mentioned a bacteria divide once in an hour. So it is divided once in an hour. So after 24 hours, it will divide 24 hour multiplied by 2 divided by it's not divided by, it is like this is number of hours they have asked and multiplied by number of division. So a bacteria is dividing 2 times per cell, so the answer will be 48. So 2 की power 48 will be the correct answer for this question. Correct answer is C. Moving on to the next question. Number of chromosome at each equatorial plate of metaphase 1 of a plant having 2n number is 60. So if 2n number is 60, if 2n number is 60, then homologous chromosome will be, and homologous chromosome pair will be 30 and that's the answer they have asked, that after number of chromosome group at equatorial plate of metaphase 1, the homologous pair, so answer will be 30. Okay, next question is, a bacterium divide every 35 minutes in a culture cell containing 10 raised to power 5, this is 10 raised to power 5 cell per milliliter in is grown for 175 minutes. What will be the cell concentration per milliliter after 175 minutes? So what we have to calculate is, calculate number of division First we have to calculate number of division. 175 divided by 35 minutes they have given. So total number of division is 5. Time for growth is given 175 and doubling time is given 35 minutes. So now we are going to calculate final cell concentration. Initial was given 10 raised to power 5. Final was given 10 raised to power 5 multiplied 2 into power 5, because number of division 2 की power n, we are using this formula. So the final answer will be 32 into 10 raised to power 5 cell per milliliter. So the correct answer is A. Next question is telomere. So what we have to remember is that telomere, they are present in eukaryotic genome only. First thing is this. Second thing, they are protecting cap that prevent end replication problem. That means, because in case of linear DNA, that is in the eukaryote, linear chromosome, what will happen that the chromosome do not, the DNA polymerase unable to polymerize the end codes of DNA. So what telomere do is form a hook shaped structure having the repetitive DNA sequences and form a hook shaped structure so that DNA polymerase is able to form the or DNA polymerase is able to polymerize that structure and it will complete the DNA polymerization. But with what will happen with the age, the telomere size get reduced and If the size of telomere getting reduced, it means the cell is getting senescent, and at the end when the number of telomere finish, the cell get die. So the correct answer is A, because it seals the end of chromosome. Okay, the next question is, number of combination of chromosome are possible in a gamete formed from a diploid cell having 10 chromosome. So we can use the same formula, 2 की power n. N is number of haploid chromosome. They have given 2n number is 10. So for the n, it will be 5. So answer will be 2 की power 5. Answer is C. Next question is NC ratio. NC ratio is nucleocytoplasmic ratio. So after cell division, what will happen to the NC ratio or nucleocytoplasmic ratio? When a cell get divided, it will form 2 or more daughter cell. So during cell division, cytoplasmic volume decreases while nucleus volume remain same, which results NC value reduces. So the correct answer is B, that NC ratio after cell division reduces. Next is congregation. Congregation is a new term. It is the bringing chromosome on equator of spindle apparatus. That means here these are centriole which are present at poles, these are astral rays, and these chromosomes are arranged at equator or equatorial plane. Okay, so correct answer is C, bringing chromosome or spindle apparatus to the equator. Next question is, if the cell number of dyad at each pole during anaphase 1 is 25, the gamete will contain how many chromosomes? Dyad here is bivalent. So he have talked about sister chromatid. So number of sister chromatids were 25. How many? A gamete will contain how many chromosomes? They have given sister chromatid 25. That means total number of chromosome will be must be 50. So after meiosis 1, it will be 25. That is why they have told us after in anaphase they are getting 25 gamete or 25 sister chromatid. So after meiosis 2, they also get 25, whether it is chromatid or chromosome. So the correct answer is A. Next question is, the undifferentiated cell of an animal has 64 chromatid. They have talked about 64 chromatid. We know that in a chromosome 2 chromatids are present. Okay, if 64, that means it is equal to 32 chromosome. If they are talking about 32 chromosome in undifferentiated cell, animal has 64 chromatid in anaphase. Metaphase, that means 32 chromosome was there. How many chromosome will be present in gamete? So we have to reduce it to the half, that means 16 in after meiosis 1, and after meiosis 2 they will remain 16. So the correct answer is D. Next question. In a culture of E. coli, the cell population increased from 1.5 10 raised to power 6 cell per milliliter to 12 into 10 raised to power 6 cell per milliliter in 36 minutes. So initial population is 1.5 into 10 raised to power 6 cell per milliliter. Final population is 12 into 10 raised to power 6 cell per milliliter. So we have to find out increase in population, which is final population divided by initial population, which is 1.5 into 10 raised to power 6 divided by 12 into 10 raised to power 6 divided by 1.5 into 10 raised to power 6 cell per milliliter. So the answer will be 8. So now we have to calculate number of generations. Number of generation, the formula is 2 की power n. So 2 की power n is given 8. So n is 3, which is 2 की power 3 will be the answer. So now we have to calculate generation time. The generation time is total time divided by number of generation, which is equal to 36 minutes, which is already given total time, divided by number of generation, which is 3. So which will be 12. So the correct answer is C, 12. Hope you have got the answer correct. If you have any doubt, do let me know. Thank you so much for your patience. Thank you so much. Next time we will try to discuss some other question. For now, thank you so much.